Hello everyone, my name is Kathy Dang and I'll be starting you all off today. We'd like to begin with introducing our presentation titled Computational Comparison of the rpsL gene in E. coli, S. pneumoniae, S. bongori, and S. fexneri. My group members are Teju, Cole, and Jack, and we'll be starting off with a brief overview of the abstract. Antibiotic resistance is a growing global health concern, and understanding the genes involved is crucial. This study focuses on the rpsL gene, which encodes the S12 ribosomal protein associated with streptomyces resistance in bacteria such as E. coli, Streptococcus pneumoniae, Salmonella bongori, and Shigella flexneri. Using NCBI's GenBank and BLAST tools, sequence comparisons revealed that highly similarity in rpsL genes were observed across the species. The E. coli strain analyzed was previously shown in studies to develop streptomycin resistance due to mutations in the rpsL, and similar sequences in the other bacteria suggest they could develop comparable resistance. Notably, E. coli shared the most highest sequence similarity with S. bongori and S. flexneri, highlighting how closely related bacteria may evolve similar resistance mechanisms. This demonstrates the value of comparative genomic tools in predicting the spread of antibiotic resistance. So our hypothesis was the rpsL gene in E. coli shows similar sequence patterns when compared to Streptococcus pneumoniae, Salmonella bongori, and Shigella flexneri. While E. coli has been exposed to streptomycin, the others have no known history of exposure. This similarity suggests that S. pneumoniae, S. bongori, and S. flexneri may also develop streptomycin resistance through mutations in the rpsL gene, much like E. coli. Now I'll pass it off to Teju.
Thank you, Kathy. Let's go through a quick background. Antibiotic resistance is one of the biggest challenges in modern medicine, and a key way bacteria becomes resistant is through mutation in ribosomal protein genes. One important gene is rpsL, which encodes for S12 protein in the 30S ribosomal subunit. This protein ensures that the protein synthesis is accurate. Streptomycin, a commonly used antibiotic, targets this part of ribosome, but in bacteria like E. coli, specific mutation in rpsL prevent the drug from binding, allowing the bacteria to survive and keep making proteins. Since ribosomal genes like rpsL are highly conserved across species, other pathogens such as Streptococcus pneumoniae, S. bongori, and S. flexneri may also have similar mutation and mechanisms of resistance. In fact, researchers have found that S. pneumonia has a version of rpsL very similar to E. coli.
So the results here show the following. Figure 2 is used to demonstrate the chunk of our values that we found using our BLAST analysis. All of our e-values for both the nucleotide and protein sequences all were less than our statistical significance value of 0.05. This means that the observed changes are highly unlikely to have occurred by chance. Moving on to our percent identities, the percent identity of pneumoniae was 72.2%. For bongori, it was 97.6%, and flexneri was 100%. That was all for the nucleotide percent identities. Percent identities for the proteins were 70.0%, 100%, and then 100% respectively to pneumoniae, bongori, and flexneri. We also displayed a graphic analysis of these results just to visually demonstrate their relative similarity. The x-axis values were arbitrarily identified as the following: 1 equaling pneumoniae, 2 equaling bongori, and 3 equaling flexneri. All percent identities were multiplied by 10 to the negative 2 just for visual cohesion. Moving on to figure 4, we have a phylogenetic analysis that shows pneumoniae is the least common ancestor, and that bongori and flexneri are most closely related. And lastly, E. coli is somewhere in between. I'm going to pass it off to Cole now.
All right, for the methods, the full nucleotide sequence for E. coli was retrieved using NCBI's GenBank and used to identify the rpsL gene encoding the 30S ribosomal protein S12. This process was then repeated for S. pneumoniae, S. bongori, and S. flexneri. Each rpsL nucleotide sequence was individually compared to E. coli using BLAST to find percent identity and E-value to assess sequence similarity and likelihood of random matches. When comparing E. coli and S. pneumoniae, the more dissimilar sequences, discontiguous megablast option was used. This option was used because these two species are not very closely related. When comparing E. coli with S. bongori and S. flexneri, the option highly similar sequences megablast was used. This option was used because these species are closely related and have very similar DNA. The 30S ribosomal protein S12 sequence for E. coli was then found, as well as the 30S ribosomal protein S12 sequences for S. pneumoniae, S. bongori, and S. flexneri. Using BLAST-P, these protein sequences were then individually compared to E. coli to find relationships in percent identity and E-value. Using this information, a phylogenetic tree was constructed to display the relationships between all bacterial strains. And I'll pass it back to Jack.
Moving on to the discussion, this study found that the rpsL gene is highly conserved when comparing E. coli with bongori and fexneri. The nucleotide percent identities for this gene were highly similar at 97.6% to 100%, with protein identities further supporting this result at 100% identity. These results indicate both a genetic and phenotypic relationship, considering that both the nucleotide and protein sequences are so similar. All E values are less than our statistical significance value of 0.05, indicating these genetic similarities are highly unlikely to be due to chance. Overall, these results suggest that there is a close genetic relationship in the rpsL gene between these species, meaning they may develop streptomycin resistance through similar means. Previous research found that point mutations in the E. coli rpsL gene are the mechanism through which streptomycin resistance was obtained. When comparing E. coli and pneumoniae, there was a significantly lower similarity. While the E-value was 1 times 10 to the negative 42nd, only 72.2% nucleotide and 70.07% protein identity was found. This indicates that the pneumoniae rpsL gene is not conserved when compared to E. coli, and alterations in the rpsL genetic sequences have occurred at some point in the past. Phylogenetic analysis further supports these results with a significantly greater distance between the nearest common ancestor for pneumoniae and E. coli, bongori, and fexneri. These findings imply that, despite being different bacterial species, the rpsL gene is highly conserved. Parallel or divergent evolution are the likely mechanism through which this conservation has occurred. This study is limited to merely demonstrating a change or similarity in genetic/protein sequence between species and cannot be used to definitively prove a mechanism through which such changes or similarities have occurred. Further study is required to prove these mechanisms, most relevant being an extended culturing experiment with all 4 bacteria from the study, each being independently exposed to streptomycin, followed by genetic analysis of the rpsL gene and further analysis for resistance. The results of this study point towards the complex challenges clinical practitioners face when considering antibiotic resistance. Other bacteria and antibiotics may also show cross-resistance, and further analysis into this phenomena may help identify potential resistance that can be made aware to clinicians in the global pursuit to prevent antibiotic resistance.